Hello and welcome to today's Wolfram Institute live stream. Today we have Willem Nielsen, a fellow at the Institute, and he'll be talking about the latest blog post with Stephen that he worked on bulk orchestration. Willem, go ahead.
Thank you, Dugan. Yeah. So, um, the post is titled "What's Special About Life: The Rulial Ensemble and Bulk Orchestration in Biology and Beyond." Just to give you guys some background, Stephen usually frames his thinking around physics. So, um, this idea of an ensemble in statistical mechanics is where you don't know the details of, a gas, let's say, but you can still derive some general properties about that gas. And those are kind of what the gas laws are. And so the question is, can we do something similar for biology where we don't know precisely the rules that generate some biological system, but can we still derive some general properties about biological systems in general as a whole? Um, and so from that, so that's that kind of set of rules. Um, so we can use the one thing we seem to know about biological systems is that they were achieved, they were evolved to achieve some purpose, right? And those rules that were evolved to achieve some purpose, we call the Rulial Ensemble. And so the question is, can we study that ensemble systematically um, for some general behavior? And what we find that's interesting is that there do seem to be general properties of the Rulial Ensemble. In particular, this idea of bulk orchestrated systems or mechanoidal behavior seems to be kind of a general property. Um, So, what we're
Yeah,
showing
so
here
if
is,
I may, yeah, so like the theme for like statistical mechanics bulk orchestration is you have like all these particles and the configuration, and then there's like some metric that you get from that, like the temperature of a system, and a lot of different configurations of position and momentum can give you the same temperature reading. And
mhm,
here in this example, like there are six CAs, cellular automatas, that have the same property at row 100 or whatever. It's like
mhm,
orange, blue, and yellow in thirds, right? And so these are like
mhm.
six different configurations of cellular automata, different rules that produce the same metric.
Exactly.
And then
Yes,
okay,
exactly.
so then
So,
like the question is, given that bulk orchestration, uh, that coarse graining, what can you say about all the different configurations that produce the same outcome?
exactly. So you have some coarse grain objective function. So in this case, for these models, we're trying to get a cellular automata that has a string of ones, a string of twos, a string of threes. And then we, do some adaptive evolution process. And then we have, different emerging solutions. And we're trying to study those solutions systematically for, similarities. So just some background on the computational model that we're using here and that we've been using for this evolution stuff is, so this is a cellular automata. If you don't know, a cellular automata starts with some initial condition. So in this case, we have a single red cell in the initial condition. We apply some rules down the page and we kind of get this resulting behavior or pattern. Um, this CA or cellular automata was found through some adaptive evolution process. So the way that process works is we take a set of rules. We take the rules of the CA and then we make some random change. And if the change leads us closer to the output sequence that we want or some objective function, which I'll explain more in the next slide, then we keep that change. If not, then we reject it. So it's very similar to how biological systems have mutations, and then certain mutations lead to higher fitness, etc. So the objective function that we're using um, in this case is, again, a string of ones in the final row, a string of twos, and then a string of threes, or a string of red, blue, and yellow. Um, And so what we're showing here is the snapshots of during the evolution, kind of the progression. Um, and so it's kind of like as evolution progresses towards some objective, what are the intermediate solutions that we get? And what's interesting is, so just backing up for a second, we start with the null rule. So we kind of start with this trivial rule, and then um, as we get closer to the solution, we get these complex solutions. Um, and then, as we get to the right solution, we get these more simpler, what we're calling kind of mechanoidal solutions. So the idea is like, again, you have these, so these ones, say this one here, it's 75 um, bits away from the correct answer. So there's 75 bits that aren't in the right place. And it has a very complex mechanism. Um, but then, as you get closer, evolution kind of locks in on this kind of simpler mechanism that generates the correct result. So now at this point, there's no bits so all the bits are in the right place. Um, and yeah, so it's kind of showing how, for this objective function at least, the kind of simpler, more mechanoidal solutions seem to do better. And um, it's an interesting result because in biology, what seems to be emerging is that, uh, you know, systems even down to the molecule scale are kind of orchestrated. So it's like this molecule hooks onto this molecule. Um, and, they're moving in very non-random ways. So
Yeah. So can you
you
explain
look like you have a question.
what's mechanoidal about this, or what do you mean by mechanoidal?
Right. So like in this rule, the yeah, the behavior looks very random.
Hmm.
Um, and we're going to kind of try to do a quantitative assessment or measure of bulk orchestration later with compression size. But for now, just qualitatively, these rules look very complex and random, whereas these rules seem to be kind of have a purposeful or non-random uh, aspect to them.
Yeah, exactly. So yeah, I see that when the loss function got to 1623, I guess, it honed in on this like pattern where you have this yellow wedge and then the purple wedge, and then in between that is like this very defined like slanted line, um, and then that kind of that persists um, afterwards. So it's like it got onto a mechanism and then keep that because it was good for the objective function.
Exactly. That's another interesting feature about these evolutions is that, they seem to have the property where, like you said, um, they're kind of related to their ancestors in a way,
Hmm.
similar to how, like, we all have the same spine as whatever, mice.
Right. Lizards and birds. I mean,
Okay.
we all have like spine, rib cage, four limbs.
Right. Right. Okay. So kind of a branch of this idea is we can take this objective function of, the number or kind of matching some horizontal final row and um, look at the kind of all possible horizontal sequences and see which sequences evolution can and cannot achieve. And one of the concepts that emerged from that is this idea of mutational complexity, which is, algorithmic complexity is the number of rules that generate a particular sequence in the entire rule space. But with mutational complexity, we're trying to say, um, the number or how easy it is for adaptive evolution to match some horizontal sequence. And specifically, the actual measure that we use is just the number of steps it takes to reach that sequence or within some threshold of that sequence.
Right.
So like,
Yeah. So
right. So, and what's interesting is when you have,
sorry, the sequence, I guess. Yeah, it's the row of like 100 colors at the bottom,
mhm,
and yeah. So I guess like what is the space of all those sequences? It's like k to the 100, 4 colors to the
right,
100, and the space of cellular automata for k equals 4, r equals 1. Um, that's considerably less. So you're not going to be able to get every sequence that you want.
Exactly. Yeah,
Uh-huh.
that's true.
Right. And then we can see here that there's a lot of overlap that given a sequence at that position, there are multiple different CAs which achieve that.
Right. So that's another important point is so you're asking to evolve to some horizontal sequence, but that doesn't necessarily mean, although you could, but you're going to find the same rule as kind of,
Hmm.
yeah. So like this one has a different behavior than this one, even though its horizontal sequence is the same.
Yeah. Yeah, that's an interesting point. I really think that this one is like really clever. Um, and this one like makes a lot of sense. And it's like it's pretty simple, but this one gets points for creativity.
Yeah, I think that's also interesting because a lot of these solutions aren't what a human would engineer, I think,
Hmm.
but it's similar to how biological systems kind of have these surprising mechanisms that wouldn't, yeah, just wouldn't be engineered, I think. Like this one, for example, it's like the, you don't need to use this blue yellow stripe thing. You could just,
Right, right. The
have yellow.
alternating one. Um,
Yeah, it's kind of like a unnecessary complexity.
yeah, this one is like more engineered, and then this one is that's the thing about life is it doesn't care like how it works. It's just if it works, we don't really care about maybe some messy details unless there's the
Yeah,
selection pressure that weeds that out.
right. Exactly. Okay, so what we can find when we kind of systematically look at these horizontal sequences, though, is that as you increase the specificity of the sequence or make it more complex, then
Mhm.
adaptive evolution has a harder time finding that solution. So, you know, this sequence is very simple, the blue and yellow, and adaptive evolution has no problem. But then in a case like this, where it's red, yellow, red, it has a much harder time finding the solution.
Right, right.
And we
Makes
can
sense.
kind of plot that. We can plot that here, showing just the sequences and then the mutational complexity or the number of steps to get within some error range of that sequence.
Okay, so this is the number of steps given evolution run.
Exactly.
Hmm.
Yeah. So, in some of the other sequences, we're not actually sure if there even exists a rule that generates that sequence. So like this one, for example, we
Uh-huh.
don't know. Um, so we can actually kind of, to control for that, you can select a rule from the space at random and then try to try to adaptively evolve to that same horizontal sequence. So you know that the rule exists, but you don't know, or you don't know if adaptive evolution can find it. And when you do that, if you choose like a simpler rule, adaptive evolution is able to do it usually.
Hmm.
But then as you kind of choose something more complex, then adaptive evolution really struggles. So it's kind of like if you're trying to search or evolve to a very specific mechanism, then adaptive evolution is going to really have trouble with that.
Okay, so this is interesting. So you took an example CA and then extracted out the bottom row of it, right? And then you
Hmm.
use that to train other ones. And in this case, it looks like it found it immediately, almost, except for there's like a pixel off up top, which is kind of interesting.
Hmm.
And so it got something basically identical um, to it, but in any
Hmm.
case, it found others that exactly replicated this guy's sequence,
Hmm.
and then this guy's bottom row is harder to find. I got pretty close, but within five, six, or seven, and this guy's row. very difficult.
Hmm.
These are very lossy. It's scoring like it's almost the row is what 100 long or 200 long?
100.
100. Okay, so they're scoring like 40% accuracy on that. Okay,
Right. Yeah. So, um,
and you're always
again, it's
adapting
like,
from you starting the evolution from the null rule each time.
mhm. Yeah. So I just kind of want to make this point though, that biological systems, they kind of have all this detail. So it's like, this protein changes shape and hooks onto this, whatever,
Yeah.
receptor. Um, and the task of biology is to try to kind of study those mechanisms and understand why they came about. But in a lot of cases, it's just kind of like, um, it couldn't be that all of those mechanisms were evolved for a particular purpose, because we're showing here that adaptive evolution actually can't evolve to systems that are that detailed. Anything that kind of it does succeed at is usually very coarse grained, and there are many ways to do that thing. Um, so it's kind of showing how, like, you know, biologists should be careful about trying to explain away every single mechanism that is in, a biological system, which is kind of the point that Stephen was making in NKS. Um, but it's kind of like
Yeah.
an even better example of it, I would say, here. And it's also like, um, not to go too far on a tangent here, but like, it's interesting that biological systems, every solution that is achieved is coarse grained, because that means something like a human brain is like, well, it seems like to us it would be very difficult to, it seems like it's a very complex kind of thing to build.
Hmm.
But in fact, it the fact that, adaptive evolution was able to, make brains, so to speak,
Hmm.
means that there's probably it's probably not that hard actually. And I guess like with LLMs and AI, we're kind of seeing that a little bit, I would say.
Yeah, I mean, in the case of the brain, it's like you have once you get started with like a little bit of processing to augment that and to get like subsystems going, different regions of the brain, make new is
Hmm.
to iterate on the base brain.
Hmm. Yeah,
This
exactly.
step by step process.
Hmm. Okay, so we can look at not just horizontal sequences, but vertical sequences. So this is a string of red cells and then blue cells in a
Hmm.
vertical sequence. Then again, adaptive evolution is able to achieve this. Um, what's nice about these is if you take, say, a periodic vertical sequence, so one red cell every 10 steps, again, it's able to find these, but then you can easily generalize and say, okay, we evolved for this particular vertical sequence of 50 steps, and we found these solutions. But what happens if we run the same solutions for 100 steps? And what's interesting is certain solutions do generalize perfectly, and then others don't. And usually the ones that are more complex uh, struggle
Hmm.
to generalize, and the ones that kind of have locked in into some kind of mechanoidal behavior um, seem to do better.
Right, right, because you're have a mechanoidal input, and
Hmm.
the chaotic ones they don't maintain. So what you evolved like this one, you evolved to match this sequence here, and then you asked it if it kept going like that, and the answer is this one didn't because it just found something that worked for that piece and didn't make something
Exactly.
that was repetitive.
Right.
Hmm.
So we can look at other objective functions as well, not just, so this one is saying just the color frequencies in the final row. So not caring about the arrangement of them, just the number of red cells, number of blue cells, number of yellow. And we're in this case looking to make that as equal as possible. Um, and the reason that these are one, all one instead of zero is because um, there's 101 cells actually in the final row, so they can't possibly be exactly equal, but
Hmm.
they're all getting as close as possible. So
Right,
this
right.
one
It's one
also
in the center, then
works.
50 on either side, and you run it from a single point for 50 steps.
Exactly.
Yeah.
Yeah, so the final row has 101. Um, right. And so we can perturb these solutions and see how it affects the frequency in the final row. Um, yeah. And so the blue arrow is just showing, uh, we change that particular cell, and then the darkened colors show all the bits that are different because of that change.
Hmm. Yeah, this is interesting because this one is very mechanoidal with those stripes, but
Hmm.
it goes random after that.
Yeah, so a lot of times the perturbation kind of awakens or like, yeah, activates rules that weren't being used before, and the rules can have very complex dynamics.
Nice, interesting. Um, all right, yeah, go on.
So um, those rules, yeah. So the question is, what happens if we evolve with the perturbation? So we, instead of just selecting a rule if it gets us closer to some objective, we're going to only select rules that get us closer to an objective given some perturbations.
Okay, so a prescribed like location where a perturbation occurs.
No, so we do like 10 random ones every
Okay,
time.
and so the objective function is the um, average densities, a target density distribution on the final
Hmm.
row, and
Hmm.
you're asking which ones are also achieving that distribution and robust to random attacks.
Exactly.
Is it like how many attacks are you using for your metric?
I actually forget this exact one. It's
Uh-huh.
anywhere between like, oh, and this one we did three.
Okay.
Yeah. In the medicine post, we went to like 5, 10,
Hmm.
but this one, yeah, we did three because it's, I don't know, we were running these for very long numbers of steps, so it's pretty intense. 5,000.
Yeah.
Okay.
Cool. So these,
So
if they were perturbed, they would still maintain their densities.
exactly. So these ones are showing cases that were robust to perturbation. Do we
Hmm.
have some examples? We should show some examples, but they, what's interesting is they're more like simple or mechanoidal than the ones that weren't robust, like these.
Hmm. Hmm. Yeah, I wonder what the mutations look like for these robust solutions.
Yeah,
Um,
we should look at that.
if the
I
perturbations
think that
still
we,
like kind of
mhm. Yeah, I know.
mechanoidal, if the perturbations were also mechanoidal, it'd make a lot of sense.
Yeah, they might have put it in the post. Maybe we can check after. Okay. So, um, even more kind of coarse grain than the frequencies in the final row, we can just ask the frequencies for the entire pattern. So this one is trying to achieve a frequency of one white cell for every 5 red cells, for every one blue, for every one yellow,
Uh-huh.
which is why they look more red.
Yeah.
And again, evolution is able to achieve. So these are all the exact frequency that we asked for. Um,
Very
and this
mechanoidal.
fitness
Yeah.
function, yeah. So again, like you have these kind of mechanisms that are biologic-like in the sense that you wouldn't engineer something like this, I think, but
Hmm.
it still works.
This one is interesting. Kind of want to. It'd be interesting to extract those rules used in the center, and then see if that does a good job at maintaining that probability distribution for the whole um, for the whole
Hmm.
critter.
Yeah, I wonder if this one generalizes. I honestly doubt it.
Okay.
Okay, so we can ask for more and more higher and higher frequency of red cells.
Hmm.
And for all of these levels of red, evolution is able to achieve the frequency perfectly. Um, so again, it's like if the fitness function is coarse grained enough, then I think adaptive evolution tends to be able to um, achieve it.
Yeah, yeah, because
And we can,
it's like a
mhm,
less specific requirement, and so it's probably a coarser grain of how many of these cellular automata in space of CAs fit
mhm,
in these broader categories.
mhm.
And so, do
Right.
they
So
do they
showing
take less time
another,
to train for, less steps?
um,
Like, are they easy to find? Let's say.
I don't know. We didn't actually plot the mutational complexity of these. For this
Hmm.
case, we did, let's see, how many? 25,000 iterations. Yeah, I mean that's about it depends on what we're comparing it to, but okay, so I'm gonna look at the another example that we had is the blue cells. So we have like in this case we're trying to evolve. So we have one white for every five blue for every one red for every one yellow, which is why they look more blue.
Hmm.
And then we're saying as we increase the number of white that we're asking for, so one white, now two white, now three white, now four white, etc. Can adaptive evolution achieve it? And again, it can. So for every level of white, these are the solutions.
So it's getting more and more sparse relative to the background.
Hmm.
Yeah, that's interesting.
Yeah. So we can look at also growth shapes. So this is saying a growth rate of one half. How close can evolution get to that? And these are the solutions for these different growth rates.
Hmm. It's interesting that there's like uh, typically a mechanoidal spine on the left or on the right, and there may or may not be a similar spine on the opposite side. So they're not symmetric in that way, but I'm trying to see if like if it's repetitive here and if it's repetitive here, but the patterns are different.
Mhm.
It seems to be very patterned on both sides, but different ones. That's very interesting.
Yeah, yeah, again, like certain different different solutions have different levels of mechanoidalness. Like this one is very um, kind of reducible.
I actually have a question.
Yeah, go ahead.
One of the things that we talked about um, ages ago in summer school, actually, Willem, was the symmetry that we see in nature and biology. And I just wonder, a lot of these solutions seem asymmetrical, and I wonder how you think about reconciling those two things.
Yeah, well, I think for it depends on the organism. Like kind of the simplest organisms, you could say like viruses. Um, yeah, I don't know. This is kind of outside of my level, but I think it depends on the biological system. Certain systems don't have symmetry, certain systems do, I would say. But I don't know. It's an interesting question.
I mean, I suppose another way that maybe these solutions are deployed are within another sort of larger balanced system. So, you know, these
Hmm.
could, yeah.
Yeah, yeah, exactly. Like animals, I feel, or like say vertebrates, were kind of all you could think of as built on top of the same blueprint. So they kind of all by default have that symmetrical, whatever, design.
Yeah, they don't float around in the ocean, so they locomote on land, and you need balance to do that
Yeah,
typically. So yeah, insects have a symmetry, especially with their legs. So do yeah, vertebrates. Um, in the ocean, I guess like the things are less symmetric, or they can be. They're just floating.
Yeah, I don't know. I sort of tend to think of even aquatic creatures as having symmetry, but I suppose that's at a level, that we can observe, and maybe down at more cellular levels, symmetry is less necessary.
yeah, for sure. Like even the fact that we're like right hand dominant, left hand dominant.
Right, right. That's that's an artifact of neurology, I guess. The way that the brain.
Yeah, our brains aren't symmetric in that sense.
Another example is like the speech center of the brain is on the left side.
Hmm.
But yeah. So anyway, these sloped cases, it's interesting to think about because like the pattern of how this is sloped. I mean, I guess if you want a slope of like 45 degrees, right? For radius equals 1, you have to think about the rule case for k00 being uh being something, right? Um, and on the right side, like you're looking at 001 or 00k going to those colors. So you have like a couple of those and a couple of those, and the pitch, the ratio is going to be what those types of rules sequence to amongst themselves.
Yes, exactly. And importantly, it's not necessarily, and we actually looked at that, it's not necessarily, so you can just look at the output frequencies of the rule. I guess this is going back to the color frequency thing,
Hmm.
but I guess it's also relevant for the growth rate. But yeah, so if you look at just the output frequencies of these rules, they won't necessarily be the correct output frequency because some of them don't get activated. So it's kind of like the difference between it's kind of like you're taking some rule and then yeah, you're using it for some purpose. But wait, so how does that relate? Oh, dang, we lost your drawing.
Oh,
How does that relate to the growth rate?
um, yeah. So it seems like um, to get something which just goes on along a diagonal, starts here and keeps going. You could say like uh, 001 outputs a 1, right? So if there was a leading
Yeah, yeah, I
0
see what you're
through,
saying.
it would repeat its own rule again once it gets there. But if you said it goes to 2, um, and
And two goes
then
to zero and
zero
then
zero two goes to 0, then you have to like put a
yeah,
0
yeah.
here, and you have to look at the 1 2 k cases and see if um, something happens there. And so what you have to do is like you start with a one and then you go to like something else that goes to something else, and then the case for 003 comes up, and then you go back to like one again.
Mm-hmm.
And so
Yeah,
this will give
exactly.
you like something. So you go down by blocks of two each time, or in this case it looks like blocks of three before it repeats. So you have like 1 to 3 on each side, or 1 to 6, or whatever the case may be. But
Mm-hmm.
you'll have a finite number of ways you can cycle through those edge cases along each side, and then you pick one that works for the left side, you pick one that works for the right side, and you have a symmetric one. I'll bet there could be ones where you say like I want a slope on the left side of something and a different slope on the right. And you
Yeah,
get a
exactly. And it's interesting too because that kind of makes sense because when you look at try to evolve to sequences that are like growing slower,
mhm.
um, or rules that are growing slower, I should say, it has a harder time, or it's harder to find these ones, I think.
Um, in the 1/10th case, can you see a cycle 2 something and a cycle 5 something?
Um, I think we can make this bigger.
That's
I'd
interesting.
have to see it for longer.
It's like they get stalled. This one definitely gets stalled here for a while this one keeps stepping, and then it looks like they kind of take steps of different lengths,
Hmm.
sort of alternate. So as long as the alternation of that averages out to be 1/10.
Hmm.
Um, right. So I guess like 1/10 would be beyond the limits of what can be achieved mechanoidally. Like I guess the mechanisms here with 4 colors is limited to sort of maybe incremental steps along each side of four.
Yeah, like these ones.
Yeah, yeah,
Like this is very mechanoidal.
right, right. And it's like one, two, whoops,
Sorry.
1, 2, 3, 1, 2, 3, 1, 2, 3, and it steps out. Same thing here, and that's a function of that. And available because the color space here is 0, 1, 2, and 3.
Hmm. Exactly. Yeah, there's a limited kind of complexity to the mechanism when the states are limited.
Mm-hmm.
Brian, did you have like is there a reason you're curious about that?
I was wondering if the thing is along the way figuring out how to factor,
I'm not sure.
or if as numbers get bigger, do composites work better than primes?
Yeah, I think we looked at like, I don't know, we should look more at that, like which precise growth rates can and cannot be achieved. I think we had a picture in the post about like rational numbers where we were looking at square root, and yeah, we looked at like 1 over pi. Okay.
In order to get irrational numbers,
Hmm.
wouldn't it want to evolve a random number generator?
Um, well, no, because like for the case of pi, the the actual growth rate, like the numbers that are the decimal points, obviously are less and less significant. So it kind of just needs to get 3.14. Yeah. So I guess maybe that's not the best thing to try. It should be more like, I don't know what would make sense then.
Well, actually, did you try getting it to evolve something that is maximizing the statistical randomness?
Yes, that it works, but it's kind of like, I mean, it just looks like a random rules. It's, yeah. Yeah, I mean, I think it's interesting that the the objective functions that like are kind of impressive to us humans are the ones that are like, they're not so simple that they can be achieved trivially, but they're not so complex like just maximizing entropy or something. That, yeah, it's kind of the ones that are in between that, and we'll kind of see later. But it's the ones that are in between that that are kind of most impressive to us humans, I think. Okay. So other objective functions, really quick, we can just look at the periodicity. So trying to evolve to some rule that has a period of 50, for example. Um, and some of these with a transient, or sorry, all of these with some of them with a transient, some not, achieve a period of 50.
Right. So it's so this one starts with like point and then it collapses back to that same point and then repeats. So this is like very exact. Um, but this one it kind of gets started and then it starts repeating,
Hmm.
something like that. Yeah, that's
And
cool that they
for
kind of
Ellie, these,
get into they get into the rhythm after a starting.
yeah, for Ellie though, these are imposed to be symmetric. So like we force them just for, and we also did asymmetric, but I think the symmetric kind of looks nicer.
Oh, you bilaterally, left and right
Hmm.
symmetry. Okay.
Yeah, it seems like if there's a good solution can that works symmetrically, it sort of is infinitely um then you've got this benefit that you can kind of infinitely repeat it.
Yeah, true. I think, yeah, I think for this one, the symmetric ones are like they do better because I think of what you're saying. Okay, um, so what we've been looking at is just kind of a bunch of examples of objective functions, but can we kind of, our original goal was to try to characterize this Rulial ensemble and see the general properties of it. So now that we kind of have intuition about what adaptive evolution can achieve and kind of the space or the bounds of that Rulial ensemble, can we, so can we kind of quantify what this mechanoidal phase or bulk orchestration actually is? So to do that, we need to compare it to random rules. So this is just showing some random rules in this rule space of k equals 4, r equals 1. And what's interesting is you kind of have these really complex rules like these ones, but then you also have these very trivial rules. And so if we, um, again, kind of make that quantitative using the compression size, for example, um, so what we're showing here is just the compression size of various rules. And so these rules that were selected randomly and are very trivial have a low compression size, as you'd expect. They're kind of more simpler. Um, and then these ones that are adaptively evolved have kind of a medium compression size. And then these ones here, again, that were selected at random or very complex, have a higher compression size. So it's kind of like we were saying, the adaptive evolution rules or the rules in the Rulial ensemble tend to be kind of in this intermediate state between very complex and very trivial.
Right. So the compression size, this is like using run length encoding or some sort of image compression scheme. Um, so anytime that there's like a wide area that's same color, you get a compression benefit. And anytime there's a repeated element, it's also you get compression there. So these are uncompressible. so to speak.
Right.
Mhm.
Exactly.
But there seems to be a bit of a circularity in the argument. You have fitness functions that fit in that place of compressibility.
Hmm. Yeah. I mean, we're evolving to, like you said, if you pick a fitness function that isn't like, for example, if you just maximize complexity, say, let's like, you can even evolve to have a higher compression size, for example, um, then yeah, you wouldn't get this result where you would just get rules that look very complex. But I think that the rules, again, that have this kind of property that they look to us humans like they're biological, or um, those ones, those tend to be uh, yeah, like the mechanoidal solutions. Does that make sense?
Um, It does, but it feels like um, th- th- I don't know if this is analogous, isomorphic, or just metaphorical. Is it looks like class 4 sits between class 2 and 3 in a similar kind of way.
Hmm. Yeah, I would agree with that.
And class 4 is the one that looks more biological because, it's isn't in the simpler, cellular automata classification cases is the things that look biological are things that are more chaotic than fully ordered and more ordered than fully chaotic.
Yeah, I think so.
And this seems like it's filtering out for that kinds of things along a different set of dimensions.
Hmm.
So the interesting ones are in the middle. Random ones are.
Yeah. Well, if Stephen had to do it over again, I don't know if he would have class 4 before rather than 3.
Mm,
Because,
good point.
or it does fit between 2 and 3, right?
Yeah.
Yeah, so it's kind of a shame that it became 4.
Well, I guess
Right.
he ordered them based on how interesting they are. So the class 4 is like on the boundary of uh, something
No,
repetitive.
I'd agree. I'd agree, but in that case, as well as in this case, they do fit logically in some kind of middle.
Yeah.
Yeah. Yeah. So we can look at the same kind of metric over the evolution process or over time. Um, and like we were seeing before, you kind of get this bump in complexity in the earlier solutions, and then as evolution kind of locks in onto some mechanism, the solution gets simpler. And it's interesting because if you look at this, so this is just showing as evolution approaches the solution, you get this higher complexity size over. So this holds over many different, um, sorry, many runs towards that same objective function. So this is just showing like the distribution of compression sizes over time. And even if you change the objective function to be um, this different sequences, you kind of still get the same bump of complexity and then kind of a little simplification.
Do you attach an interpretation to that effect?
Yeah, I think it's because it's kind of easy to find complex rules. So if you start at the trivial null rule, you're going to find early solutions are going to be very complicated because those are just easier to find. And then slowly it's going to get closer to the solutions that are more correct, but also simpler.
Yeah. Um, that's interesting. That they go through a period of like getting complex and they sort of hack away at that and then get something which is simpler. And that's like,
Hmm.
huh. I wonder if there's a correlation between um, the complexity curve and the um, the fitness curve.
I mean, that's this.
Oh, I see.
Mhm. Okay, so just to conclude, again, what we saw with many different objective functions is that the ones that are computationally bounded or, coarse grain, um, evolution is able to achieve those objectives. And once we kind of establish that, set of rules that achieve some purpose, or the Rulial Ensemble, we can see how those rules tend to be more mechanoidal or bulk orchestrated. Um, And so, in Stephen's words, the work here provides the raw material for something we've never had before, a well-defined and general fundamental theory for the operation of living systems. It won't describe all the historical accident details, but I'm hopeful that it will provide a useful global view of what's going on that can potentially be harvested for answers to all sorts of useful questions about the remarkable phenomenon we call life. So, thank you guys.
Excellent
That's
work.
it.
Are there any other questions on chat? Um, Thank you again, Willem. Thanks for everyone tuning in. We will see you again next time.